Hey guys, so the past few weeks we've been developing an understanding of the various metabolic pathways necessary to synthesize pretty much every important material in our body, such as ATP, energy molecules, proteins, fats, uh, or lipids, from various food sources available to us. So today we're going to kind of tie it all in together to talk about one of the most important functions, which is generating nucleotides for DNA production and RNA production. Because as we know, our body doesn't just melt down the macronutrients like carbohydrates, fats, and proteins directly into energy and new tissue. The nutrients have to be broken down through a series of interconnected, complicated biochemical pathways in which sugar and fats can be used to create energy molecules that our body is capable of using for normal bodily functions such as cellular respiration or regeneration, and in turn DNA replication. And then here's just an example of the process laid out from start to finish, from glucose being synthesized into glucose 6-phosphate, and then going into the glycolysis pathway, which will feed into the citric acid cycle, and then the electron transport chain, basically is synthesizing all the ATP the bulk amount of ATP our body is going to produce, as well as essential lipid synthesis, like here, like cholesterol, all the way to basically glucose being a precursor, all the way to the complicated proteins and nucleotides, so protein synthesis and nucleotides necessary for making DNA. So let's get into it. The long essential process for forming nucleotides will begin, as well as ATP, will begin with glucose, and for nucleotides, we'll begin with the activation of ribose 5-phosphate acting as the template. So here we can see ribulose 5-phosphate being synthesized from glucose 6-phosphate into ribose 5-phosphate, and then we'll get back to this a little bit more, but it's going to basically create the PRPP template for nucleotide synthesis. But as we've talked about, glucose is also an essential molecule for forming ATP production, so the main energy molecule in our body. So I made a 9-minute video about this already, this complicated process, and if it looks complicated and confusing, it's because it is. But also because this picture isn't very detailed. But because we've already familiarized ourselves with these pathways and cycles, I don't feel the need to go into too much incredible amount of detail. So I'm just going to kind of lay out where each step starts or occurs at the cellular level, and what the main function is, and like necessary product that it produces. So beginning with the early steps of glycolysis here, it's going to happen in the cytosol of the cell. We're going to be using ATP molecules that have previously been formed or generated. They're basically going to react and form the glucose ring with several specialized enzymes, and it's going to phosphorylate the glucose. And then NAD plus will be reduced to NADH, and then that will form 1,3-bisphosphoglycerate. Enolase will occur here, and then we'll basically get the 3-carbon pyruvate molecule formed from glycolysis. Will react in the pyruvate dehydrogenase complex, again using ATP and NAD plus molecules and specific enzymes to get the 2-carbon acetyl-CoA, which will basically be sent into the 2 molecules of that will be sent into the citric acid cycle, reacting with oxyl acetate from yet another metabolic pathway to form citrate, and then the process, again using ATP and NADH molecules, they'll form NADH and FADH molecules, which are going to be used for the most important step of ATP production in the electron transport chain happening in the inner mitochondrial membrane. It's a series of electron transporters acting basically as a proton pump, an electron transporter, to shuttle electrons from NADH and NADH2 or FADH2 to molecular oxygen, and then in the process, protons are pumped into the mitochondrial matrix, into the inner mitochondrial membrane space, and then ATP synthase can occur. So ATP synthase is basically a biomechanical motor that it's an enzyme that sits in the phospholipid bilayer of the mitochondria and acts as a hydrogen ion portal. Here we can see the hydrogen ions on the outside, and they're basically going to move down the concentration gradient, but as they do, they're going to churn or turn this little motor, basically, and so we have ADP molecules and phosphate groups entering the protein here, and basically when the hydrogen ions enter down the concentration gradient, they're going to produce the bulk of the ATP molecules. So like we outlined before, about like 30 ATP molecules are going to be produced from 30 to 36 are going to be produced from this cycle as a whole. And ATP is a nucleoside triphosphate with a ribose sugar, adenine base, and 3 phosphate groups, where a high energy bond attaches to the 3rd phosphate group to the molecule. This bond is highly unstable, and that's why we get so much energy from it. In addition to that energy, ATP also is essential for nucleotide production, which we will talk about now. So what are nucleotides? Nucleotide is composed of a phosphate group here, a sugar group, pentose sugar ring here, and a nitrogen base, and they are synthesized using PRPP, or phosphoribosylphosphate pyrophosphate, which is a product of the pentose phosphate pathway that we kind of outlined here. Basically, the ribose 5-phosphate is synthesized from glucose 6-phosphate and gets an ATP attached to the ribose phosphate by ribose phosphate pyrophosphokinase. So here's the blue comes from the ribose, and the red comes from the ATP molecule. So the nucleoside production is kind of outlined here. Here's just kind of the general overview of the pyrimidine ring being synthesized from bicarbonate and orditate. So the nucleoside production, which is just a nucleotide without a phosphate group yet, is outlined here. So here's the general overview of the pyrimidine ring synthesis and purine ring synthesis, which we'll get into a little bit more. But here we can see like PRP being added to these reactions and basically forming IMP here and UMP here. So pyrimidine synthesis will start with ATP being added to bicarbonate and carboxyphosphate, and then glutamine from another biopathway will make carbamic acid. And then from there, the amino acid aspartate will be added to carbyl phosphate to produce carbyl aspartate. Aspartate. And then a hydrogen ion gets added to carbyl aspartate, forming some water, and then dihydroorditate. And then NAD plus will be reduced to NADH to give us orditate. And then from there, orditate can be added to PRPP, the molecule we were talking about before, to give us the last intermediate in forming urdolate. And then here we can see UMP, ATP being added to UMP and kinase to give us this molecule here, where we get UTP, which can further be synthesized with glutamine and water to give us CTP, as well as an inorganic phosphate group. UMP is also synthesized into TTP with the help of thiamidylate synthase. It is turned into TDP down here, and then an ATP is added, basically giving us the ring structure here, the proper ring structure here. And then purine ring synthesis, which also you can see it's pretty complicated and don't have like a ton of time to go into each individual step of each reaction, but basically IMP is the precursor for the purine nucleotides and synthesized through this big long reaction, basically PRPP loses the PP part, the 2-phosphate group, here the pyrophosphate, and the byproducts from another metabolic pathway, ammonia and glutamine, are going to turn PRPP into 5-phosphoribosyl 1-amine, we can see here. And then through this reaction sequence, we're going to end up with IMP, which is inositol. Inositol 5-monophosphate. So then IMP can then further be further synthesized to either AMP, adenylate, to form ATP, or GMP, guanylate, to form GTP. So with the help of IMP dehydrogenase and GMP synthase, we can form GMP using ATP and an inorganic phosphate group. And then similarly, with AMP, we can form ATP using GTP as the energy molecule, as well as a inorganic phosphate group to get ATP. And so basically, the purine and pyrimidine bases are going to serve as the hydrogen bonding sites for DNA and RNA, which we'll get into now. So basically, the newly formed ribonucleotides are going to serve as the building blocks of RNA and DNA. So as we know, RNA is a little bit different than DNA. It consists of cytosine, guanine, adenine, and uracil, whereas the uracil in DNA is replaced with thymine. But again, basically through another complicated process, the ribonucleotides will be reduced by ribonucleotide reductase and NADPH to form the nucleoside monophosphates called the deoxyadenylate, deoxyguanylate, deoxyacetylate, and thymidylate, which will serve as the building blocks for the deoxyribonucleic acid that we all know and love, with RNA serving as the information carrier for DNA synthesis.